Top 1. Ancient tuberculosis. In 2008, during the excavation of a Neolithic burial site near the eastern Mediterranean, archaeologists uncovered a skeleton unlike the others in the trench. The spine was sharply curved forward, with several vertebrae partially collapsed and fused together. At first glance, the deformity appeared traumatic, perhaps the result of a fall or blunt force, but laboratory imaging told a different story. Microscopic analysis of the vertebral bodies revealed characteristic lytic lesions consistent with Pott's disease, a severe manifestation of tuberculosis. Genetic sequencing later confirmed fragments of ancient Mycobacterium tuberculosis DNA embedded in the bone matrix. The pathogen had been there for thousands of years, silent but preserved. This was not an isolated case. Similar spinal collapses have been identified in remains from Africa, Europe, and the Americas, some dating back more than 9,000 years. The damage follows a recognizable pattern. The bacteria settle in the lungs, then spread through the bloodstream, attacking the vertebrae. Over months or years, the infection erodes bone from within. The spine bends, the rib cage compresses, breathing becomes shallow and painful. Eventually, the body weakens under chronic inflammation and internal bleeding. Unlike sudden epidemics, tuberculosis does not strike quickly. It lingers. Victims may survive for years while coughing blood into the soil of their settlements. For early human communities with no understanding of bacteria or airborne transmission, the illness would have seemed almost supernatural. A healthy hunter gradually wasting away, skin pale, shoulders hunched, breath rattling in his chest. No visible wound, no predator, just slow deterioration. What complicates the picture is evidence of prolonged survival. Several skeletons show advanced vertebral damage, yet the bone around the lesions had partially healed. This indicates the individuals lived months or even years after the infection became severe. They were not abandoned immediately. Someone fed them. Someone carried water. Someone allowed them to remain within the group despite the risk. Tuberculosis spreads through respiratory droplets. In enclosed shelters or cave dwellings, transmission would have been almost inevitable, yet early humans continued to cluster for warmth and protection. Close proximity meant survival against predators, but it also meant shared air, shared infection. Modern researchers estimate that latent TB can remain dormant in the body for decades before activation. If that biological trait existed in ancient strains, entire populations may have carried the bacteria unknowingly. Periodic outbreaks could erupt when food became scarce or stress weakened immune systems. Climate shifts, migration, and nutritional decline would have amplified vulnerability. In several burial grounds, individuals with advanced spinal lesions were interred carefully, sometimes accompanied by tools or ornaments. They were not discarded. They were remembered. But no written record explains what their communities believed was happening. Did they attribute the wasting illness to spirits, curses, or unseen forces in the air? Did they notice patterns of spread within households? Or did each case feel like an isolated tragedy? Today, tuberculosis remains one of the deadliest infectious diseases in the world. Its genetic lineage stretches back deep into prehistory, possibly evolving alongside early Homo sapiens as they migrated out of Africa. Some evolutionary biologists propose that TB adapted to human population density, becoming more virulent as settlements grew larger. If so, the disease was not just a companion of civilization, it may have shaped it. The skeletons remain silent. Their curved spines tell of prolonged suffering, of air shared in close quarters, of a pathogen older than cities. If tuberculosis was already embedded in human communities before agriculture fully emerged, then the idea that disease followed civilization may be incomplete. It may have traveled with us from the very beginning. Top 2. Prehistoric leprosy. In 2009, while examining skeletal remains from a 4,000-year-old burial site in western India, osteologists identified a skull marked by a distinctive pattern of erosion around the nasal aperture. The upper jaw showed resorption. The front teeth had loosened long before death. The bridge of the nose appeared collapsed inward. At first glance, it resembled severe trauma. But under magnification, the bone told a different story. The damage followed a recognizable biological signature consistent with Hansen's disease, commonly known as leprosy. Further testing confirmed the presence of ancient Mycobacterium leprae DNA within the bone. The bacterium that would later become synonymous with exile and stigma had already been infecting humans thousands of years before biblical references or medieval quarantines. This was not a disease of recorded history, it was prehistoric. Leprosy does not kill quickly. It attacks peripheral nerves, gradually numbing extremities. Fingers lose sensation. Minor injuries go unnoticed. Infections set in. Over years, facial bones begin to deteriorate. The nasal cavity collapses. The voice changes. The body becomes visibly altered. In tightly bonded hunter-gatherer groups, such transformation would not have gone unnoticed. Unlike tuberculosis, which hides within the lungs, leprosy announces itself slowly but visibly. The face changes. The hands weaken. Skin lesions form. In small prehistoric communities where survival depended on physical strength and group cooperation, such deformity may have carried profound social consequences. Yet burial evidence complicates assumptions. At several ancient sites across Asia and Europe, individuals with skeletal signs of leprosy were buried with care. Some were placed in central areas of cemeteries rather than isolated peripheries. Grave goods have been found beside them, suggesting they retained social identity. This challenges the later historical narrative that leprosy inevitably led to exile. In its earliest presence, the disease may not yet have carried the stigma it would acquire centuries later. Transmission patterns remain debated. Leprosy spreads primarily through prolonged, close contact, often via respiratory droplets. In prehistoric conditions, long-term exposure within family groups could allow the bacterium to pass quietly between individuals. Because symptoms can take years to appear, early humans would have struggled to link cause and effect. A child might develop deformities long after contact with an infected relative. The illness would appear spontaneous, unpredictable. Ancient DNA studies indicate that multiple strains of M. leprae were already circulating across continents by the Bronze Age. This suggests migration played a role in dissemination. As human populations expanded trade routes and settled in denser communities, the bacterium traveled with them. It did not require large cities to survive. It only required time and proximity. The psychological impact is difficult to reconstruct, but not impossible to imagine. A visible transformation of the face carries deep symbolic weight in human societies. Recognition, identity, and emotion are read through facial expression. When bone structure changes, when speech shifts, when sensation fades from hands and feet, the individual becomes altered not only physically, but socially. Some anthropologists propose that early ritual practices surrounding purification or spiritual cleansing may have roots in attempts to explain such chronic conditions. Without germ theory, a slowly deforming body would appear cursed, marked, or touched by unseen forces. Yet the archaeological record does not consistently show systematic isolation during prehistoric periods. In several communities, care appears to have continued. Leprosy's incubation period can exceed 5 years. In that span, an infected individual would continue contributing to the group, unaware of the silent bacterium reshaping nerve tissue beneath the skin. When symptoms finally emerged, it would be too late to reverse the damage. If prehistoric communities witnessed gradual facial collapse and nerve paralysis without any visible injury or predator, what explanation could they have formed? Was the illness perceived as punishment, inheritance, or fate? And if stigma later became attached to leprosy, did it originate not from biology, but from fear of the unexplained? The bones suggest the disease was present long before written laws of exile existed. What remains uncertain is whether the fear came first or whether fear was learned over generations as deformity became impossible to ignore. Top 3. The zoonotic brucellosis shift. In 1984, during an excavation of a 6,500-year-old farming settlement in what is now central Turkey, archaeologists uncovered a cluster of adult skeletons buried beneath the floor of a mud brick dwelling. Several of the bones showed unusual surface pitting around the lower spine and sacroiliac joints. At first, the lesions were attributed to general wear from agricultural labor, but detailed microscopic analysis revealed chronic inflammation patterns inconsistent with simple mechanical stress. The joint damage was symmetrical. The erosion extended deep into the vertebral margins. When fragments of bone were tested for microbial residues, researchers detected traces consistent with Brucella bacteria. Brucellosis is a zoonotic infection, meaning it spreads from animals to humans. Today, it is commonly transmitted through unpasteurized milk, direct contact with livestock, or exposure to infected animal tissue. The presence of the bacterium in a Neolithic farming community suggested something profound. Disease was no longer arriving solely from the wild. It was emerging from domestication itself. Brucellosis does not usually kill swiftly. Instead, it produces prolonged fever, severe joint pain, chronic fatigue, and intermittent relapses that can last for years. In early agrarian societies, where daily labor determined survival, such symptoms would have been debilitating. An infected herder might appear healthy one week, then bedridden the next. Fevers would spike unpredictably. Pain in the hips and spine would make lifting grain or tending animals nearly impossible. The archaeological evidence shows that as humans transitioned from nomadic hunting to permanent settlement, skeletal markers of chronic joint inflammation increased significantly. This shift aligns with the domestication of goats, sheep, and cattle. By living in close quarters with animals, early farmers created a new ecological bridge. Bacteria that once circulated primarily among livestock now had direct access to human hosts. Unlike tuberculosis or leprosy, brucellosis originates across species boundaries. It represents a turning point in human disease ecology. The moment humans chose to corral animals, milk them, and share enclosed living spaces, they altered the microbial landscape around them. Every enclosure became a potential reservoir. Every birth of livestock carried unseen risk. In the settlement where the infected skeletons were found, animal bones were stacked within a few feet of sleeping areas. Dung was used as fuel and building material. Ventilation was minimal. Under such conditions, airborne particles and contaminated surfaces would have facilitated transmission. Yet no written warning, no ancient tablet recorded the danger. The connection between livestock and fever would have remained invisible. Stable isotope analysis indicates that dairy consumption was increasing during this period. The introduction of milk into the human diet provided nutritional advantages, but also exposure to pathogens. Without pasteurization, Brucella bacteria could survive ingestion. The body would respond with cycles of inflammation that appeared mysterious and inconsistent. Several skeletons from the site display signs of long-term survival despite advanced joint damage. Like tuberculosis cases, these individuals were not immediately discarded. They were buried beneath homes, integrated into domestic space even in death. This suggests that chronic illness did not automatically sever social ties, but it also indicates that infection may have spread quietly within households. Modern brucellosis can persist in bone tissue and relapse decades after initial infection. If ancient strains behaved similarly, entire communities may have experienced recurring waves of fever without understanding their source. The transition to agriculture is often framed as progress, but in biological terms, it marked the beginning of sustained exposure to animal-borne disease. As more ancient DNA studies are conducted, evidence continues to mount that many modern zoonotic diseases have prehistoric origins. Brucellosis may have been among the earliest to establish a permanent foothold in human populations following domestication. If the rise of farming enabled civilization, it also created the conditions for new pathogens to flourish. The question is not whether early humans invited these bacteria into their settlements. The question is whether they had any choice once domestication began. Top 4. Ancient treponemal disease. In 1997, during the excavation of a prehistoric burial mound in the American Midwest, archaeologists uncovered a skeleton whose long bones bore a strange and unmistakable pattern. The tibia was thickened and bowed outward. The surface of the skull displayed crater-like depressions radiating across the frontal bone. These were not battle wounds. They were not animal damage. Under magnification, the lesions followed a repetitive and symmetrical formation known in forensic pathology as caries sicca. The pattern pointed toward a treponemal infection, a family of diseases caused by spiral-shaped bacteria in the genus Treponema. Today, the most infamous member of that family is syphilis. But treponema diseases include several variants, such as yaws and endemic syphilis, which can spread through non-sexual skin contact in warm, humid environments. Radiocarbon dating placed the skeleton more than 3,000 years before European contact with the Americas. This detail matters. For centuries, historians debated whether syphilis originated in the New World and was carried to Europe by Columbus's crew, or whether it already existed in the Old World long before transatlantic voyages. The bones do not settle the debate, but they complicate it. Treponemal infections progress in stages. In early phases, skin lesions may appear and disappear. In later stages, the bacteria attack bone. The tibia thickens. The skull develops destructive pits. Over years, the face and limbs can deform subtly but permanently. Unlike leprosy, treponemal diseases do not usually numb extremities. Instead, they inflame tissue from within. In multiple prehistoric sites across Africa, Asia, and the Americas, similar skeletal damage has been documented. The consistency of the lesions suggests these infections were not rare anomalies. They were persistent. Some skeletons show advanced bone remodeling, indicating individuals survived long enough for the body to attempt repair. The mode of transmission in prehistoric populations remains uncertain. If early strains resembled yaws, the disease could have spread through simple skin contact among children or within families. In humid tropical regions, this would have allowed rapid circulation without requiring sexual transmission. Over centuries, bacterial evolution may have altered virulence and pathways of spread. The social impact is difficult to reconstruct, but the physical evidence is undeniable. Bowed legs would alter gait. Cranial lesions would change facial contours. In small groups dependent on hunting, farming, or gathering, even minor physical impairment could reduce productivity. Yet burial practices show no uniform pattern of exclusion. Many infected individuals were interred within communal graves. One particularly revealing case from a prehistoric European site showed both treponemal lesions and signs of healed fractures unrelated to infection. The individual had survived multiple health crises. This suggests ongoing care within the group. Chronic illness did not always equal abandonment. Modern molecular analysis has identified ancient strains of treponema in skeletal remains predating written history. Genetic comparison reveals divergence points suggesting these bacteria have been adapting to human hosts for thousands of years. They did not appear suddenly in medieval Europe. They were already embedded within human populations in varying forms. If treponemal diseases were circulating globally in different variants long before recorded pandemics, then the narrative of sudden emergence may be incomplete. The bacterium may have traveled alongside early migrations, shifting form as environments changed. Unlike tuberculosis or brucellosis, treponemal infections leave distinct and visible signatures in bone. They carve patterns that remain long after flesh has vanished. Those patterns speak of prolonged infection, of bacterial persistence, of adaptation across generations. If ancient communities observed these deformities without understanding bacterial causation, what interpretation did they form? Was the illness perceived as inherited fate, environmental curse, or something passed silently between bodies? The bones suggest a pathogen that evolved with humanity, altering itself as societies change. The unanswered question is whether modern forms are merely the latest chapter of an infection that has never truly disappeared. Top 5. Prehistoric parasitic worms. In 1972, archaeologists excavating a rock shelter in the American Southwest uncovered what appeared to be compacted fragments of dried soil lodged near an ancient hearth. At first glance, the material seemed unremarkable, but laboratory testing revealed it was coprolite, fossilized human feces, preserved for nearly 8,000 years in the arid climate. Under a microscope, the sample transformed from dust into a biological archive. Embedded within were dozens of oval-shaped structures with thick outer shells. They were unmistakable. Ancient parasite eggs. Subsequent analysis identified species consistent with roundworms and whipworms, intestinal parasites that today infect millions in regions lacking modern sanitation. These organisms live within the human digestive tract, feeding on nutrients meant for their host. They cause chronic abdominal pain, anemia, weakness, and in severe cases, intestinal blockage. Unlike sudden epidemics, parasitic infections operate quietly. They persist. Additional coprolite samples from South America, Africa, and Asia revealed similar findings. Trichuris tricura eggs, Ascaris lumbricoides eggs. In some samples, larval hookworms were detected. The evidence was geographically widespread and chronologically deep. Human hosts had been carrying intestinal parasites for thousands of years before written language emerged. The implications are stark. In prehistoric settlements without plumbing, waste disposal systems, or understanding of microscopic life, contamination would have been constant. Soil near sleeping areas could become infected. Water sources downstream from encampments could harbor parasite larvae. Children playing barefoot in contaminated ground would be especially vulnerable. Unlike tuberculosis or treponemal disease, parasitic worms rarely leave marks on bone. They leave no visible lesion after death. Their impact is physiological rather than skeletal. This makes coprolite analysis crucial. Without preserved fecal matter, their presence would remain invisible in the archaeological record. Chronic parasitic infection can lead to iron deficiency anemia. Symptoms include fatigue, pale skin, and reduced cognitive function. In hunter-gatherer societies, diminished stamina would directly affect hunting efficiency and food-gathering capacity. In agricultural communities, it would limit labor output during planting and harvest. Over time, widespread infection could suppress entire populations subtly but persistently. Some coprolite samples contain not only parasite eggs but also pollen and plant residues, revealing diet. In several cases, researchers observed heavy parasite loads alongside nutritional deficiencies. This suggests a biological feedback loop. Poor nutrition weakens immune response, increasing parasite survival. Parasites then exacerbate malnutrition by siphoning nutrients. Hookworms, in particular, penetrate the skin and migrate through the bloodstream before settling in the intestines. In tropical prehistoric environments, bare feet would have provided easy entry points. A single infected individual could contaminate soil that remained infectious for weeks. Without footwear or hygiene practices, reinfection would have been constant. What makes parasitic infection uniquely insidious is its normality. A child born into such an environment would never know what an uninfected body felt like. Chronic fatigue would seem ordinary. Occasional stomach pain would be part of daily life. Entire communities may have functioned under a baseline of reduced physical capacity without realizing it. In one remarkable case from a 5,000-year-old Andean site, coprolite analysis revealed evidence of deliberate consumption of medicinal plants with known anti-parasitic properties. This suggests that prehistoric humans may have observed correlations between certain plants and symptom relief, even without understanding the underlying biology. Parasites represent a form of coexistence. They rarely kill immediately. They exploit. They endure. Their evolutionary relationship with humans may stretch back tens of thousands of years. If early humans carried invisible organisms within them from birth, organisms that drained energy and nutrients without obvious external signs, how would they interpret recurring weakness? Would it be seen as personal frailty, spiritual imbalance, or environmental curse? Unlike the dramatic collapse of a spine or the visible erosion of a skull, parasitic infection leaves no dramatic skeletal warning. It hides in the soil and in the gut. And for much of human prehistory, it may have been the most common illness of all. Top 6 Early Smallpox Strain. In 2016, researchers analyzing ancient human remains from Northern Europe extracted fragments of viral DNA from teeth dated to approximately 3,000 years ago. The genetic material did not belong to bacteria. It matched sequences related to variola virus, the pathogen responsible for smallpox. The finding unsettled long-standing assumptions. Smallpox had been widely believed to emerge in recognizable form during historical periods recorded by Egyptian and later Roman sources. But the genomic fragments suggested an ancestral strain circulating far earlier than written accounts. Unlike tuberculosis or brucellosis, smallpox does not hide quietly within bone. It erupts across the skin. High fever strikes first, followed by waves of pustular lesions that spread from face to limbs. In severe cases, mortality can exceed 30%. Survivors are left scarred, sometimes blinded. In prehistoric communities with no immunological understanding and no isolation protocols, such a disease would have been catastrophic. Skeletal remains rarely preserve clear evidence of smallpox because the virus primarily affects soft tissue. However, dental pulp can trap viral DNA fragments in certain burial conditions. In several Bronze Age individuals, genetic signatures indicated a now extinct lineage of variola that differed from later historic strains. This ancestral variant may have been less virulent or transmitted differently, but its existence confirms the virus's deep antiquity. Transmission of smallpox occurs through respiratory droplets and direct contact with lesions. In densely settled prehistoric villages, particularly those transitioning to agriculture, close quarters would have facilitated rapid spread. Unlike parasites that linger for years, smallpox moves fast. Entire households could become infected within days. Burial patterns in some late prehistoric cemeteries show abrupt increases in mortality over short time spans. Clusters of individuals interred simultaneously without signs of trauma raise the possibility of epidemic disease. While definitive diagnosis is difficult without preserved viral DNA in every case, the demographic signatures align with acute outbreaks rather than slow chronic illness. Modern genetic reconstruction indicates that smallpox evolved through a series of mutations over thousands of years. The Bronze Age strains appear genetically distinct from those that ravaged medieval Europe and later the Americas. This suggests a long evolutionary pathway, with earlier forms potentially circulating at lower mortality rates before becoming more lethal. For early human communities, the sudden appearance of fever, rash, and disfigurement would have defied explanation. A person could be healthy one week and covered in lesions the next. Children would be especially vulnerable. Survivors, marked permanently by facial scars, would carry visible reminders of the outbreak long after it passed. The virus requires a human host to survive. It does not persist long in the environment. This means prehistoric outbreaks would have depended on continuous chains of transmission within migrating or interacting groups. As populations grew and trade routes expanded, viral spread would have intensified. Unlike tuberculosis, which can remain dormant, smallpox either kills or confers immunity. Survivors become resistant. This biological dynamic can shape population structure. Over generations, selective pressure may have influenced genetic resistance within certain communities. The discovery of ancient variola DNA complicates the timeline of epidemic disease. It suggests that humanity's confrontation with viral pandemics began long before recorded plagues. The absence of soft tissue evidence in older remains does not prove absence of infection. It may only reflect preservation limits. If smallpox or its ancestral variants were already circulating in prehistoric settlements, then the concept of first pandemics shifts backward in time. The visible scars left on survivors would have been interpreted without scientific context. Were they seen as marks of survival, divine selection, or contamination? The genome fragments recovered from Bronze Age teeth are incomplete, yet they whisper of a virus evolving silently alongside early civilization. The question is not whether smallpox existed in prehistory. The question is how many outbreaks occurred without leaving a written trace, erased except for faint molecular echoes locked inside bone. Top 7 Prehistoric Waterborne Epidemic. In 1993, excavators working at a late Neolithic settlement near a dried river basin in southeastern Europe uncovered an unusual burial pit. Unlike the orderly graves found elsewhere at the site, this trench contained multiple bodies layered rapidly, some positioned face down, others partially overlapping. There were no signs of blunt force trauma, no embedded projectile points, no defensive wounds. The skeletons belonged to men, women, and children alike. Radiocarbon analysis indicated they had died within a narrow window of time, possibly weeks apart. The absence of violence shifted suspicion toward disease. But which disease leaves no clear mark on bone and kills swiftly enough to overwhelm a settlement? Modern epidemiologists studying similar demographic patterns point to acute waterborne infections. Pathogens such as Vibrio cholerae, certain strains of Escherichia coli, or other enteric bacteria can spread rapidly through contaminated water supplies. Severe diarrhea leads to dehydration. In extreme cases, death can occur within hours. Unlike tuberculosis or brucellosis, which erode the body slowly, waterborne outbreaks strike explosively. In prehistoric communities dependent on shared wells, rivers, or irrigation channels, a single contaminated source could infect dozens simultaneously. Without knowledge of microbial contamination, the connection between water and illness would have remained invisible. At the Neolithic site, sediment samples taken from the former riverbed revealed elevated levels of fecal bacteria residues. While specific pathogens cannot be conclusively identified thousands of years later, the environmental context suggests the settlement relied heavily on the nearby water source. Seasonal drought followed by flooding could have concentrated waste upstream before washing it into drinking supplies. Burial evidence strengthens the epidemic hypothesis. The bodies were interred quickly, with minimal grave goods and little ritual spacing. In ordinary circumstances, prehistoric burials show care in positioning and ornament placement. Here, urgency replaced ceremony. Several skeletons displayed contracted positions consistent with rapid death during dehydration and muscle cramping. Acute gastrointestinal infections caused severe fluid loss, sunken eyes, and muscle spasms. Victims weaken rapidly. In tightly clustered dwellings, the disease would move from one family to another in days. Fear would spread alongside infection. Unlike viral diseases that confer immunity, certain bacterial waterborne illnesses can reappear seasonally if sanitation conditions remain unchanged. In early settlements lacking sewage separation, contamination would be cyclical. Heavy rainfall could flush waste into communal sources. Drought could concentrate pathogens in stagnant pools. Isotopic analysis of the individuals in the mass grave revealed no significant nutritional deficiency compared to earlier burials. This suggests the deaths were not due to famine. Instead, they appear sudden and widespread. In other prehistoric sites across Asia and the Middle East, archaeologists have documented similar burial clusters near ancient water systems. While definitive microbial identification remains rare due to DNA degradation, the repeated pattern of rapid, nonviolent mortality near water sources raises a consistent possibility. The rise of permanent settlements introduced new vulnerabilities. Waste accumulated. Human and animal excrement gathered near dwellings. Rivers once used by nomadic groups only briefly were now relied upon continuously. Stagnation replaced movement. For early humans, the concept that clear water could carry invisible danger would have been counterintuitive. Water is life. It sustains crops, livestock, and families. To suspect it as a source of death would require an understanding of contamination that would not emerge for millennia. If one season's flood transformed a village's lifeline into a vector of lethal infection, how would survivors interpret the catastrophe? A curse on the river, anger from unseen forces, punishment from the sky. The trench remained silent, its hurried burials marking a moment when an unseen agent swept through without leaving scars on bone. The true pathogen may never be identified, but the demographic imprint suggests a pattern that would repeat throughout human history. Top 8. Ancient hepatitis. In 2018, researchers analyzing ancient DNA extracted from human teeth discovered fragments of a virus embedded deep within the dental pulp of individuals who had died nearly 7,000 years ago. The burial sites spanned parts of Europe and Western Asia. The viral sequences did not belong to bacteria. They matched hepatitis B virus, a pathogen that today infects hundreds of millions worldwide. The discovery forced a revision of medical timelines. This was not a recent disease, it was prehistoric. Hepatitis B does not deform bone. It does not leave visible skeletal scars like tuberculosis or treponema disease. It attacks the liver, an organ that vanishes long before archaeologists arrive. That makes it uniquely dangerous in the prehistoric record. Without molecular analysis, its presence would remain completely invisible. Genomic reconstruction revealed something even more unsettling. The Neolithic strains were genetically distinct from modern variants. Some lineages appear to have gone extinct entirely. Others evolved into forms still circulating today. This suggests a long evolutionary relationship between humans and the virus, stretching back thousands of years. It also implies that entire viral branches may have risen and disappeared without documentation. Transmission pathways in prehistoric contexts likely included blood exposure, close contact, and possibly childbirth. In tightly clustered Neolithic settlements where tools were shared, minor injuries common, and ritual practices involved contact with bodily fluids, opportunities for viral spread would have been frequent. Unlike smallpox, hepatitis B can remain silent for years. An infected individual may show no symptoms while unknowingly transmitting the virus to others. Chronic hepatitis B can lead to liver inflammation, cirrhosis, and eventually liver failure. The progression is slow. Fatigue, abdominal pain, and jaundice would emerge gradually. In a prehistoric community, these symptoms might be interpreted as weakness, poisoning, or spiritual imbalance. The connection between one sick individual and another months later would be nearly impossible to recognize. Unlike plague or cholera, hepatitis B does not produce sudden mass graves. It erodes health over decades. That makes it biologically strategic. A virus that does not kill quickly has more time to replicate and spread. In small prehistoric populations, such a trait increases long-term survival of the pathogen. Stable isotope data from Neolithic skeletons show increasing population density associated with farming settlements. Permanent housing meant repeated contact between the same individuals over years. This ecological stability benefits viruses capable of chronic infection. Each birth provided a potential new host. Vertical transmission from mother to child may have allowed the virus to embed itself within family lines for generations. The discovery of ancient hepatitis B DNA also revealed recombination events, genetic exchanges that shaped the virus's evolution. Some prehistoric strains share features with modern African and Asian genotypes, suggesting ancient migration routes carried the virus across continents. Human expansion spread not only culture and agriculture, but pathogens encoded at the molecular level. The absence of skeletal damage makes hepatitis B archaeologically elusive. Entire communities could have carried chronic infection without leaving a single visible marker in bone. Only advanced sequencing technology uncovered its presence. This raises a deeper implication. If one silent virus persisted undetected for millennia, how many others circulated beyond our current capacity to identify? In prehistoric life, there would have been no awareness of liver inflammation or viral replication. Only unexplained fatigue, swelling, and sudden decline in adulthood. A hunter slowing earlier than expected. A mother weakening after childbirth. A respected elder succumbing without visible injury. Hepatitis B represents a different kind of prehistoric terror. Not explosive, not visibly deforming, but embedded within bloodlines, evolving quietly as humanity transitioned from nomadic movement to settled existence. If viruses like hepatitis B were already evolving alongside early farmers thousands of years ago, how many modern diseases are simply the latest forms of infections that have always been with us? Top 9. Prehistoric plague strain. In 2015, geneticists examining dental pulp extracted from a 4,900-year-old skeleton in Central Asia detected fragments of bacterial DNA that would redefine assumptions about one of history's most infamous killers. The sequences matched Yersinia pestis, the bacterium responsible for plague. Until recently, plague was primarily associated with medieval pandemics, such as the Black Death. But the genetic signature embedded within that prehistoric tooth suggested a much older origin. The burial itself showed no signs of violent trauma. The skeleton was part of a small cluster of individuals interred within a narrow time frame. Radiocarbon dating indicated they lived during the late Neolithic to early Bronze Age transition. Further genomic analysis revealed that this early strain of Y. pestis lacked certain genetic adaptations that later enabled efficient transmission via fleas. This detail is crucial. It suggests the bacterium existed in human populations long before it evolved into the form that would devastate medieval Europe. Plague in its bubonic form is transmitted primarily by flea bites carried by rodents. But early strains appear to have spread through direct contact or respiratory droplets, more akin to pneumonic plague. Without the flea vector mechanism, transmission would have required closer human-to-human interaction. In compact prehistoric settlements, that condition would have been easily met. Unlike rickets or brucellosis, plague can move rapidly through a population. High fever, swollen lymph nodes, septicemia, and organ failure can follow within days. Mortality rates can exceed 50% without intervention. In early human communities with no antibiotics or quarantine protocols, even a less virulent ancestral strain could have caused devastating mortality clusters. Additional ancient DNA findings from sites in northern Europe and Siberia have identified similar early plague strains dating back more than 5,000 years. The distribution pattern aligns with migration routes associated with steppe populations expanding westward during the Bronze Age. This has led some researchers to hypothesize that plague may have accompanied large-scale human migrations, silently shaping demographic shifts. Unlike tuberculosis, which often leaves skeletal lesions, plague typically kills too quickly to mark bone. Its archaeological footprint is therefore subtle. Mass graves lacking signs of violence, demographic imbalances skewed toward sudden mortality, and now genomic traces preserved in teeth provide the primary evidence. One striking aspect of early plague genomes is the absence of the gene that allows the bacterium to block a flea's digestive tract, a key mechanism behind explosive medieval outbreaks. This suggests prehistoric plague may have been less efficient at causing widespread epidemics, but still lethal in localized clusters. Over centuries, genetic mutations enhanced transmission capability, transforming a regional pathogen into a global catastrophe. For prehistoric observers, the sudden onset of high fever followed by rapid death would have been terrifying. Unlike chronic diseases that unfolded slowly, plague would strike without warning. A healthy individual could fall ill and die within days. No visible wound, no predator, no drought, just abrupt collapse. The presence of Y. pestis DNA in Neolithic remains forces a reconsideration of disease timelines. Plague was not a medieval invention. It was already embedded within human populations thousands of years earlier, evolving quietly. If early strains circulated intermittently across prehistoric settlements, they may have contributed to unexplained population declines long before recorded pandemics. Migration waves might have been partially driven by invisible microbial pressures rather than solely climate or conflict. The bacterium that would later decimate cities may have been refining its strategy across millennia, adapting alongside its human hosts. The genetic fragments retrieved from ancient teeth serve as molecular fossils, proof that plague's history is deeper than once believed. If Yersinia pestis walked beside early humanity long before chronicles existed, how many prehistoric communities vanished under its influence without leaving more than scattered bones and silent genetic traces? Top 10. Unknown prehistoric epidemic. In 2002, during construction work near a prehistoric settlement site in Eastern Europe, machinery exposed a pit containing more than 20 skeletons layered tightly together. The bodies were positioned irregularly, some overlapping, some partially flexed, with no consistent burial orientation. There were no embedded arrowheads, no defensive fractures, no burn marks. The individuals ranged from small children to elderly adults. Radiocarbon analysis revealed they died within a narrow span of months approximately 6,000 years ago. Unlike other burial grounds at the same site, which displayed orderly placement and ceremonial goods, this trench bore signs of urgency. Soil fill appeared hastily compacted. Grave goods were absent. The dead had been placed quickly and covered without extended ritual preparation. The pattern suggested sudden, collective mortality. Initial hypotheses considered famine or environmental collapse. However, isotopic testing of bone collagen showed no prolonged nutritional deficiency leading up to death. The individuals were not chronically starved. Their skeletal development, particularly in children, indicated relatively stable food intake prior to the event. Violence was ruled out through forensic analysis. Trauma markers were absent. Researchers turned to the possibility of epidemic disease. Ancient DNA extraction was attempted from dental pulp and petrous bone, the densest part of the skull known to preserve genetic material. The results were inconclusive. No clear signature of known pathogens such as Yersinia pestis, Mycobacterium tuberculosis, or Brucella species emerged. This absence does not imply absence of infection. Viral pathogens degrade rapidly and rarely leave recoverable DNA after millennia. Certain hemorrhagic fevers, respiratory viruses, or rapidly lethal bacterial strains may leave no skeletal trace and no surviving genetic fragments. The degradation of nucleic acids over thousands of years means many ancient diseases remain undetectable. What remains is the demographic imprint: simultaneous deaths across age groups, rapid burial without evidence of ritual delay, settlement abandonment shortly after the event, indicated by layers of windblown sediment over domestic structures. It appears the survivors, if any, left. Comparable sites across Europe, the Near East, and parts of Asia reveal similar mass graves dating to transitional periods between hunter-gatherer and agricultural lifeways. In several cases, pathogen testing has failed to identify a definitive cause. Yet the mortality patterns suggest contagious spread rather than environmental disaster. Modern epidemiology recognizes numerous pathogens capable of causing swift, high-mortality outbreaks that leave minimal skeletal evidence. Influenza-like viruses, hemorrhagic fevers, certain strains of meningitis, and waterborne toxins could eliminate populations in weeks. Without immunity, isolation, or medical intervention, prehistoric communities would have been defenseless. The archaeological record preserves only fragments of these events. A trench, bones, silence. Unlike plague, tuberculosis, or treponemal disease, which leave identifiable biological fingerprints, some pathogens vanish entirely after their outbreak passes. They may have evolved, mutated, or gone extinct. In several of these mass burial sites, there is no clear continuation of occupation immediately afterward. The land was left unused for generations. This suggests not just mortality, but fear. If an invisible agent swept through a settlement, killing indiscriminately, survivors may have abandoned the location entirely, associating it with danger beyond comprehension. The inability to identify a specific pathogen underscores a deeper uncertainty. The prehistoric world likely hosted microbial threats that no longer exist in recognizable form. Evolution eliminates some organisms while amplifying others. Entire disease lineages may have risen and disappeared without leaving more than skeletal clusters behind. If unknown pathogens once erased villages before written language could record them, how many chapters of human suffering remain undocumented? The bones do not speak the name of the agent that killed them. They only mark the aftermath. These prehistoric diseases reveal a pattern that is far more unsettling than isolated illness. They suggest that from the very beginning of human settlement, invisible biological forces were shaping survival, migration, and even social structure. Tuberculosis, bending spines in silence. Leprosy, reshaping faces without explanation. Zoonotic bacteria emerging the moment humans chose to domesticate animals. Viral strains evolving long before history could name them. Entire communities collapsing near water sources. Generations of children growing with weakened bones as agriculture altered diet and sunlight exposure. And mass graves where the pathogen left no trace except silence. This is not a sequence of unrelated afflictions. It is a biological continuum. As humans clustered, settled, traded, and migrated, pathogens adapted beside them. Each shift in lifestyle created new ecological niches. Each technological advancement introduced unintended microbial consequences. Disease was not merely an obstacle to early civilization. It was a constant evolutionary pressure that shaped it. There is no evidence of coordinated suppression in these ancient cases, yet there is a different form of erasure. The vast majority of prehistoric outbreaks left no written record. Viral genomes degraded, bacterial DNA fragmented. Only fragments of bone remain. Modern science reconstructs what it can, but countless microbial lineages have likely disappeared without identification. Entire epidemics may have occurred, reshaping populations and migration routes, without leaving more than demographic anomalies in burial sites. What was lost with each vanished pathogen is not only life, but knowledge. Early humans faced diseases without understanding causation, transmission, or immunity. Their interpretations were shaped by fear, observation, and survival instinct. Rituals, taboos, and social boundaries may have emerged partly in response to patterns they could sense, but not explain. The threat is not confined to the past. Pathogens continue to evolve. Zoonotic spillover remains possible. Genetic mutation never stops. The same biological mechanisms that haunted prehistoric settlements still operate. The difference is awareness and intervention, not immunity from emergence. The question is not whether early humans were terrified by disease. The question is how many times unseen microbes redirected the course of human history before we even understood what they were. If these ancient chronicles of survival and silent pathogens have changed the way you look at the history of our ancestors, please subscribe and like the channel. We are digging deep into the history that time tried to bury. Join us as we uncover the secrets of the past to understand our future. The next discovery is waiting just beneath the surface,